we'll talk about reproductive phenology in Australia rainforest plants, which species where, when, and maybe even why. Let's welcome him.
So I'm just going to be talking about some research that I'm doing as part of my PhD. And it's mainly focused in subtropical rainforests of Australia. Okay, so in Australia there's a long history of colonisation and removal and exploitation of the Indigenous people. So I just want to start my talk by acknowledging that the traditional owners on the land where I work. So my research takes place on the land of these people that I've listed here on the slide. So you may have spent time in forests and seen some of these amazing sites where the ground is completely covered in fruits or the canopy is just a patchwork of beautiful colour. And if you haven't seen it, then you should come to Australia and have a look. It's really beautiful. So there are a lot of animals that are specialised to pollinate and disperse these trees, but as the climate changes, some of these species might start to fruit more often, or perhaps never again. So if we don't understand what drives trees to flower and fruit, then we won't know how rainforests are going to change into the future. So these sites and ideas inspire the questions that my research hopes to answer. Alright, so I'll just give you a brief introduction at the start, and then I'm going to take you through different parts of my research. So trees produce flowers and fruit for recruitment, and the timing of this is known as reproductive phenology. So this is linked to local and larger scale biotic and abiotic environments. For example, a really common relationship in rainforests are where animals are provided with a food resource in return for pollinating flowers and dispersing seeds. However, trees depend on pollinators and dispersers being around at the right time, and those animals also depend on the tree phenology linking up with their life cycles. So, for example, Australian fruit doves that feed on rainforest fruits, they nest and produce young during times of peak food availability. And all of this is really important for maintaining ecological networks that influence the recruitment and the restoration of our future forests. So phenology of both plants and animals can be cued by different things. Tree phenology in many cases is cued by temperature and rainfall, whereas animal phenology has been linked to photoperiod. So like I mentioned earlier, plant-animal interactions rely on these cues lining up, but they are at risk if they start to become misaligned. Some misalignments are being caused by climate change. Many plants are starting to flower, fruit, and produce leaves earlier than normal in response to warming climates. And animals are struggling to align their life cycles with these changes. So that is the case for the European skylark. It migrates thousands of kilometres to its breeding ground, and it relies on oak tree leaves emerging so that they can feed their young. But the birds are starting to arrive too late to access peak food. Other mismatches can come from fauna declines. So back in Australia, connections have been made between the loss of large frugivorous birds like the southern cassowary and reduced genetic diversity of trees that produce really big fruits. So one way that we can describe tree phenology is to classify it into patterns by looking at how often species reproduce. So we could see a continuous reproductive pattern where trees reproduce multiple times in a year. And this might be because flower availability has to align with the life cycle of a highly specialist pollinator. Or we might observe that trees with fleshy fruits are reproducing annually. And this is perhaps because they need to maintain generalist animal dispersers. And lastly, we might observe the superannual pattern where reproduction is occurring with big gaps. And this may be because trees that are wind pollinated need to synchronize their flowering for pollination, or trees that are wind dispersed don't need to maintain those animal dispersers, and in fact they might be maximising the recruitment of seedlings by starving and then overwhelming their seed predators with synchronised fruiting. So here are some examples of what reproductive patterns look like within forests. So these figures show the proportion of individual trees or species that have those different reproductive patterns that I was just talking about. And subannual here means that trees reproduce 2 times in a year. So if we look at this top from La Selva in Costa Rica, population studies have found that most tree species are reproducing one or more times in a year. And these patterns were linked to predictable temperature and rainfall cycles. In this bottom figure, we see species patterns from a rainforest in Southeast Asia and Lambi Hills. And here there is a superannual reproduction pattern which becomes the most common. So interestingly, these forests are dominated by wind-dispersed species, and studies have found that these species synchronize their reproduction, perhaps to overwhelm seed predators, and these events are also being cued by global climate oscillations like the El Niño event. So when we look to measure these patterns, people have traditionally used seed traps and direct observations, which are labour-intensive, but they are pretty accurate. But increasingly, we have these novel data streams that are being incorporated into phenology research. So these include historical herbarium specimens and citizen science observations, and these can provide information on when and where a species was seen flowering and fruiting. Now, I'll just quickly bring these methods into the global context. So this map shows the locations of detailed field studies that have contributed to one global database of flowering and fruiting phenology, but Australia's contributions are really sparse. So there are 32 Australia is here. There are 32 Australian species represented in this data set, but there are only 2 rainforest species. So I've been talking about Australian rainforests, so I'll just give you a bit more background. So rainforests in Australia cover a continental gradient from these strong seasonal dry forests in the Northern Territory up the north. And then in northern Queensland, we get these subtropical rainforests, tropical wet rainforests in Cairns and subtropical rainforests in Brisbane, all the way down to temperate rainforests in the south. So about 1/3 of these fall within UNESCO World Heritage Areas, but since colonisation, which was 200 years ago, about 75% have been cleared, which leaves only 2% of all native forests in Australia being rainforests. However, these areas provide habitat for over half of all plant and butterfly species in Australia, 40% of bird species, and 35% of mammals. So rainforest trees show a phenomenal amount of variation, not only in how they grow, but how they reproduce. So to illustrate this, here are the fruits and seeds from around 300 Australian species, and it's not only really beautiful to look at, but this suggests that there may be different reproductive patterns. So climate change influences phenology, and both climate and other local environments, as well as pollination and dispersal strategies of trees, influence their phenology. Yet we lack this fundamental knowledge for many Australian rainforest species. And we need this information to provide a more complete picture of how phenology is linked to animals and the environment. So this is what my research focuses on. So hopefully at this point you're primed with the knowledge of why reproductive phenology is important to understand, but that we don't know yet where Australian phenology fits into the global context. So I'll just go into a bit more detail. So we really don't know that much about reproductive phenology, or at least that's the initial impression that we get when we look within the global context. Are species reproducing like those forests in Costa Rica, or are they more like the forests of Southeast Asia? So to answer this question, I did 2 things. Firstly was a systematic lit review, and I tried to find all the unpublished phenology studies as well. And I also identified any novel data sources that might be available. So this allowed me to work out how people are studying reproductive phenology, what types of plants are being focused on, where and for how long research is taking place. And the second thing I did was to look at novel data sources. So like herbarium specimens, and I compared these to field data. And I'm going to show some preliminary results of this approach for the fruiting patterns of an animal dispersed and a wind dispersed species. So what I found is that there is phenology data in Australia that comes from many sources. However, these vary in their spatial and temporal coverage, accessibility, and we don't yet know how good some of these are for representing phenology patterns. Sorry, there's supposed to be colour coordination, but I think this got a bit lost in the email. So I found a few studies from both published and unpublished sources, and generally these had short monitoring periods and only captured a few species or locations, and none of them considered any seed trap data. So I also found that we can source data from seed collectors science observations that are available online. So these offer significantly larger spatial and temporal coverage, but we don't know yet if we can use them to represent patterns. Okay, so getting into some more details, there are 7 rainforest-specific papers in Australia, and they have a really big range of monitoring times. There are 2 papers that captured the most number of species, which is this one here for the wet tropics and this one from the subtropics. But they only looked at the month of flowering, they didn't look at patterns. So we also found 3 unpublished phenology studies that cover the wet tropics and the subtropics. And I'll just give you a little example. So this study has been undertaken by a botanist called Bill McDonald. This is Bill here measuring a tree. And he's been monitoring around 40 species with monthly records of the intensity of flowering and fruiting since 1998. And here he's been taking binocular observations, scoring the intensity of reproduction on a scale of 0 to 5 being intense flowering and fruiting. So I've also found data from native nurseries who collect seeds for restoration projects. And this is likely going to be a useful source of information, but we haven't been able to identify any more yet. Okay, so herbarium specimens, I found that over the last 200 years, what you can't see there is that there are over 8 million specimens that have been collected, most of which are available online. But these haven't been used yet to infer phenology patterns in Australia. So I'll just quickly take you through how herbarium specimens can be turned into phenology observations, and then we'll look at how these compare to high-resolution field data. So I turned the intensity observations from the field study that I showed earlier into a binary where we just had a 0 if there was no reproduction or a 1 if there was flowering or fruiting. Then for those same species, I downloaded the herbarium data from 1998, and I just applied a spatial buffer to select species in a 200-kilometre radius of our field study. And then I turned those observations into a binary where any herbarium specimen that had flowers or fruits was a binary score of a 1 that species was flowering. So I'll just briefly show you the results from 2 species. So here are the results of a fleshy fruited species. The y-axis is just yes or no if that species was fruiting in that year, and the x-axis is the years of our study. So in almost every year of monitoring, we recorded this species as fruiting. However, here are the fruiting patterns of that same species if we just looked at the herbarium data. But we also need to remember that the herbarium data represents presence only. So I'll just highlight 2 ways that this high-resolution field data differs from herbarium specimens. So in this situation, where the field study has observed fruiting, but there are no corresponding fruiting herbarium specimens, it's likely that there were just so few herbarium specimens that it's not that the species didn't fruit, it's just that people didn't report it. However, when we get the opposite, and herbarium specimens are fruiting in years where the field study didn't observe fruits, this could only be because the observations are coming from 9 individual trees. So here are the results of a wind-dispersed species. Again, y-axis, yes or no fruiting, x-axis is years. So this species has really dramatic mass fruiting and flowering events, and the field observations reflect that in more of a superannual pattern. Now, interestingly, we only had 1 herbarium specimen from this time period, and we couldn't increase this if we increased the spatial and temporal range of our sampling. So these preliminary results suggest that there are options for combining data sources for increasing the spatial and temporal coverage, but that I need to do more to understand how these data can be used together. I'm going to skip over some bits because I don't have too much time left, but just lastly, we're going to turn this into a database of reproductive phenology. So there's lots of data in the world, but having this all spread out makes it difficult to discover and use. And centralized databases have really advanced all fields of science, which includes phenology. The Master Tree Plus is the name of that global database that I showed earlier. And this is a database of counts of flowers and fruits. Now, other types of databases are also being developed. Closer to home, in Australia, we have Wild Obs, which is run by a researcher called Matthew Luskin, which I know a lot of you may know. And there's also terms phenocams. which looks at phenology data of leafing out of trees. So what I've found is that there is phenology data in many locations in Australia, but they come from many sources, and considering them all together is quite difficult. So what I'm going to do is I'm going to create a database of phenology observations, and this is going to be pulling all of these different sources together into a centralized location so it can be easily assessed, and then we can use it to answer more questions. So this is just showing you an example of what the data source will look like. It's just going to be a binary yes/no score of flowering or fruiting. There'll be a species, a location, a date, and then where the data was sourced from. How much time have I got left?
Over already.
I'm done? Okay, I'll stop there. If you want to know more about my research, you can come talk to me later. Okay, thank you.
Thank you very much. So we are finishing up the presentation by the young researcher student. We will have a lunch break until 1 o'clock. Like I said earlier, after afternoon session, we go directly to the common room, the field. So you need to change your clothes and get ready. So after we have the two lecturers, and then we go directly. So after lunch,
Are we going on a long walk?
It's about 2 hours walk around, but it's not very steep. So it's like walking, you know. We'll have in the other mountains a hiking, very, very hard on Friday. But today it's not too hard, you know, we can just walk around. So don't worry too much, but you need to have a long sleeve and the trousers, make sure you're protected, okay? And get ready, okay? Have a nice lunch and see you at 1 o'clock.